we may take that the backbone of primary structures. Backbone of primary structures. Now here, amino acids, you know that is alpha amino acids. So that here R group, that is alpha carbon, and COOH group, and NH2 group is attached with alpha carbon, and here H and that R. Now we may take that here N, and here C, and here H and R, and C double bond O. Then it is one amino acid sequence. It is dependent on R group. It is dependent on R group. If R group is hydrogen, then it is glycine. R group is CH3, then it is alanine. Likewise, we may classify all those 20 amino acids. Now, another one, it is NH. It is NH and C, and here H and R, and here C double bond O, and here N and C, H and R, and C double bond O, and here NH, and here NH. This is the backbone of all amino acids or proteins. So here one amino acid and another one amino acid. So it will be gives a sequence. So primary structure represents a sequence. It is one amino acid and another one amino acids and another one amino acids. And here we may take that the secondary structure. Now, the secondary structure depends on these R groups. The secondary structure depends on R groups. Now, here in a primary structure, the functional group, what is it? Here NH, NH2 is amino group, and C double bond O group is a functional group. And these functional groups are exposed. It is exposed outwards. Is exposed outwards surface. So it is exposed in outwards, so that these functional groups interact each other and make hydrogen bond or sulfur, it is disulfide bond. So the NH and the CO functional groups are exposed outwards, so that there is interact with each other. These two are interact with each other and making a hydrogen bond. Now, this hydrogen bond and disulfide bond makes the primary structure get coiled or twisted. So, this hydrogen bond or disulfide bond makes primary structure get coiled. Because of hydrogen bond and disulfide bond. And this coiling may be two types. It is alpha helix and beta flat or beta pleated. So, we are taking a secondary structure, that is making a hydrogen bond and a disulfide between the functional group because they are exposed in outwards. Now, Because of this hydrogen bond and disulfide bond, coiling is possible, and the coiling may be two types. One is alpha helix and beta pleated. Now, we are going to alpha helix structure. So, it is alpha helix structure. So, in this secondary structure, alpha helix structure. And the alpha helix is nothing but the R group is larger. So, R group size is larger, there is a possibility of alpha helix. And here there is R and CH, NH2 and COOH. Now, the R group size is larger, then it is alpha helix structure. The protein is alpha helix structure. Now, it is a spiral structure. Spiral structure are coiled into right hand screw. Right-handed screw. Right-handed screw. So, it will be coiled into right-handed screw or spiral structures. This alpha helix structure is otherwise known as, 3.613 helix. 3.613. What is the meaning of 3.613 helix? And here it will be coiled like this, right-handed coil. Okay. Now, each turn, that is, each turn contains 3.6 amino acids. 3.6 amino acids. 3.6 amino acids, and the NH group of one amino acid, one NH group makes hydrogen bond to CO group, to the CO group of third amino acids. Third amino acids. It is nothing but 1,3 in fraction. And also it is 13-membered ring is formed. 13-membered ring is formed. So that it is called as 3.613 helix. 3.6 amino acids in each turn. Each turn contains 3.6 amino acids, and the hydrogen bond between, that is 1 and the third amino acids, and it has 13-membered ring. So it is alpha helix structure. And example, that is keratin and wool and silk. These are all alpha helix structure. Alpha helix structure. Now, we may take beta pleated structure. Beta flats or beta pleated. And here it is also depending upon the R group size. The size of the R group is smaller, then it is beta flat. Then the size is moderate, then it is beta pleated structure. So depending upon the size, it may be a beta flat or beta pleated. In this beta pleated structure, beta pleated structure, each group is in a zigzag manner. Each polypeptide is in zigzag manner. Now, suppose here each group makes hydrogen bonded with two other neighboring groups. Each polypeptide chain make hydrogen bond with two neighboring group. Suppose here the polypeptide chain, here NH, N, C, and here H and R, and C double bond O, here NH, and it is again N and C, H and R, C double bond O, N and H, C, H and R, and here C double bond O like this. It is one polypeptide chain. And one more as the same. What is that? Here it is NH and C, and like this, that is R and C double bond O, N and C, H and R, C double bond O, and NH and C, H and R and C double bond O, like this. Now one more. Here it is NH and C, H and R group, C double bond O, and here it is NH, that is a hydrogen, and C, H and R and C double bond O, and NH and C, H and R and C double bond O. Now it is the structure. Now we may take one polypeptide. One polypeptide chain is making a hydrogen bond with adjacent to two neighbors. Now here this making a hydrogen with adjacent neighbors. So this is a hydrogen bond. One polypeptide chain making a hydrogen bond with another one. The same way. Here there will be a hydrogen and making that a hydrogen bond. And here NH, this is making a hydrogen bond. It is making a hydrogen bond. So here we may take that one polypeptide chain making hydrogen bond with two other neighbors. And this may be a parallel. What is a parallel? We may take like this. Here it is a parallel one and making a hydrogen bond. Suppose here NH and here C double bond O, that is making a hydrogen bond. It may be a parallel. It may be a parallel or anti-parallel. Anti-parallel. So anti-parallel means here zigzag manner, it is opposite one. This is anti-parallel. So beta pleated structure is parallel or anti-parallel way. Okay, now we may take that. That is a tertiary structure. Tertiary structure. And this tertiary structure represents overall folding of polypeptide chain. So it represents overall folding. Overall folding. Overall folding of polypeptide chain in three-dimensional space. Three-dimensional space. Then it represents tertiary structure. Okay. Now, this gives rise to two shapes. This folding gives two types of shapes. One is fibrous. And another one, it is globular. So, tertiary structure represents the fibrous protein and the globular protein. If it is a fibrous, it may be a rod or rope-like structures. Then, if it is a globular, then it is a spherical structure. So, depending upon the tertiary structure, we are classifying the protein into fibrous and globular. We are already seen that is essential and non-essential amino acids, and we are taking that is depending upon that functional group, acidic and basic, and the neutral amino acids. And one more classification, it is fibrous and globular. So it is a protein classification, it is fibrous and globular. Okay, so the overall folding of polypeptide chain that represents, that is tertiary structure. Okay, now it is the quaternary structure. Quaternary structure. And many proteins can exist in a single polypeptide chain, but certain cases the protein may composed of two or more polypeptide chain. So we may take many proteins are single polypeptide chain. Single polypeptide chain. But in certain cases, in certain proteins, two or more polypeptide chain presence. And so these proteins are composed of two or more polypeptide chain. They are called as subunits. And these subunits are may be identical or different. It may be an identical one or different. Okay. So here the quaternary structure refers the subunits. So quaternary structure refers number of subunits, and its spatial arrangement. For example, we are taking a hemoglobin. Hemoglobin have 4 polypeptide chain as a subunits. So we are taking a hemoglobin as 4 subunits. So this is a primary, secondary, and tertiary and quaternary structure. So structure of protein, first primary structure represents the sequence of amino acids, and sequence changed, then biological function is changed. Then secondary structure is represents, that is a folding chain by alpha helix and beta pleated by the size of R group. And tertiary structure, the overall folding in a three-dimensional way. Then quaternary structure represents subunit and spatial arrangement. Suppose if the protein has primary structure, suppose we are taking the primary structure. If it is a primary, then it will be coiled, normal coil, and it is secondary. Then it will be get folded. It is get folded, then it is a tertiary structure. And more subunits. Here one subunits, and here one subunits, and one more subunits, like this, then it is the quaternary structure. So it is the primary and secondary and tertiary structure of proteins. Now we may take denaturation process. It is denaturation of proteins. And you know that proteins are unique three-dimensional structure and specific biological activity. Now, the laws of biological activity is known as denaturation. So, denaturation refers laws of biological activity. Biological activity. Now, on denaturation, there is only the laws of secondary and tertiary structure, and primary structure of protein is retained. So, this denaturation is done by one is maybe increasing the temperature, or heating action of proteins. For example, egg albumin is converted into coagulated, and it may be done by changing of pH. The third one, by using organic solvent. So organic solvent is also denatured. The protein get denatured by organic solvent. And fourth one, presence of alkali or acids and some salts. It is also protein get denatured in presence of alkali, acids and salts. And presence of urea. So urea also added to protein get denatured. And detergents. So protein get denatured by detergents like, that is dodecyl hydrogen sulfate. Okay. Now, test for proteins. The first test is Biuret test. In this test, we are taking the reagent. It is copper sulfate. in presence of sodium hydroxide. Now we are taking a protein by adding the copper sulfate in presence of sodium hydroxide, that is basic medium. The blue color of copper sulfate is changes into violet color. Blue color is changes into violet color, so that we may identify which contains proteins. Then second one, it is xanthoprotic test. Xanthoprotic test. The reagent is concentrated nitric acid. Now, protein with concentrated nitric acid, the color changes become orange or yellow. Suppose the nitric acid is touched by our hand, it is nothing but a skin, is a protein, and the skin become yellow or orange color in presence of nitric acid. So, protein with concentrated nitric acid produces a yellow or orange color. Yellow or orange color. The third one, it is ninhydrin test. What is that ninhydrin? It is cyclic triketone. Cyclic triketone. It is OH and OH, double bond O and double bond O, is hydrated cyclic triketone. Protein in presence of ninhydrin gives purple in color. Purple color. So by that way we may identify the test for protein. One is biuret test, xanthoprotic test and ninhydrin test. Okay, thank you.